This is my Wolbachia final presentation. Wolbachia is an intracellular bacteria that mainly impacts insects. It infects 20% of all arthropods on Earth, like all insects. It affects a species population by increasing its female population by infecting them. Overall, it reduces the population, especially males, because there's not many females giving birth to insects. But for humans, it leads to sicknesses like elephantiasis and African river blindness. But on the plus side, it also decreases our chance of being bitten by mosquitoes because there's less females giving birth. Our campus is participating because we are studying this bacteria because we want to study the interaction of the different species and more about their evolution, ecology, developmental biology, etc. And it's important for students because it's meant to help them think critically and solve problems more effectively. This is my webflow, my project flowchart. We showed up for these labs at least once a month. This is my collection data. I collected it a few months ago at night, just after sunset, at my backyard in my house. I found it. I found it in the grass, like while I was outside, and I used an empty water bottle and a coffee cup to like capture the fly. Flies or dipteras. Sorry. Their habitat is mostly garbage cans, plants, pet areas, and soil. Their diet consists of a variety of liquids, decaying fruits and vegetables, food waste, and poop. Their life cycle is between a week to 3 weeks, so 8 to 25 days. Their unique traits are they vomit on human food and then suck up the solution so they can gain nutrients. And they in the process they contaminate the food or whatever they're feeding on. This is my infection statistics from literature. The prevalence in my journal 1 is 21%, and that was from Malaysia. The climate was humid, and it was found in a tree branch. The second article had a prevalence of 62%, and its phenotype was cytoplasmic incompatibility. It was found in Turkey. The temperature was mild. It was winter, and the other temperature was hot, and the climate was dry summer. Natural breeding sites were unavailable on the website. My third article was, it had a 17% prevalence. It was found on campus, and the climate was humid during the summer. My initial prediction was that it would be absent, that Wolbachia would be present or would be absent inside the insect after PCR testing, because based on the tear samples from 2013 to 15 in the two articles, it wasn't likely that my insect was going to be infected with Wolbachia, especially for flies. Um, for the lab, my quality was clean. It came out clean because it was above a 1.7 nanogram per milliliter, and proteins and other contaminants were filtered out. These are my calculations for the absorbances and credit absorbances and concentration. This was my expected result, or my prediction. As you can see, there wasn't a I wasn't predicting a Wolbachia gene in it, but this is the actual result, and it resulted in a positive. So I found that there was a Wolbachia gene present in my diphtheria sample, and for the electrophoresis experiment, 100 volts were used and 2% agarose gel was used. Yeah. Oh, and the Wolbachia was located at 708 base pairs, and my sample was located at 438 base pairs. This was the class data that my prediction was based on. My initial prediction of my diphtheria was not being infected, and it was contradicted by data results from the last slide. And my prediction was based on this info of that diphtheria had a 20% prevalence of infection from fall 2018 through spring 2020. One benefit I can reflect on this lab is the appreciation of the work of scientists and doctors do on researching COVID and other diseases on a molecular level. I'm glad that I now have an idea on how to use PCR that's used in actual studies and research labs for viruses and more. I feel that this lab has been one of my more relevant courses I've ever had in Georgia Southern. And that's it. And here's my references.